So let's review what we know so far. We know that we have a couple of very important ions inside and outside of the cell, a lipid bilayer membrane, and some ion-selective leakage channels. We know that when we measure the resting potential in a neuron, we're measuring an electrical gradient, or the difference in potential energy between the inside and the outside of the cell. We know that an electrical gradient forms across the membrane because of the leakage of ions down their concentration gradient. We know that the fluid inside and outside the cell is basically net electroneutral, and that only a very small number of ions like potassium moving across the membrane can cause big changes in the potential across the membrane. And most importantly, we know that an equilibrium is reached when the concentration gradient and the electrical gradient are equal and opposite. So we've hopefully developed an intuitive understanding of how concentration gradients and selective ion channels give rise to a membrane potential. But we can also precisely predict and calculate the membrane potential if we know the concentrations of the ions and a few properties of the membrane. If you have a system with just one ion that can pass through the membrane, and if you know the internal and external concentrations of that ion, then you can calculate what the electrical potential would be that would balance out the concentration gradient at equilibrium. We can do this using something called the Nernst equation. This equilibrium potential tells us at what membrane potential electrical forces and diffusion exactly balance each other out. Let's look at the different components of the Nernst equation so that we can understand it better. The variable E on the left side of the equation is the dependent variable that we're solving for. This variable may be referred to by multiple names that you'll hear throughout neuroscience courses, sometimes with subtle differences in their meaning. We're going to try and explain these differences now so that we can use these terms precisely. There are three terms that can all refer to the variable E: Nernst potential, reversal potential, and equilibrium potential. When we solve for the Nernst equation for a given ion and its concentration inside and outside of the cell, we're solving for the point at which the electrostatic force exactly balances out the diffusional force. In solving the Nernst equation, we arrive at this balancing point, which is called the Nernst potential. The Nernst potential is always calculated for a single ion species, and is usually denoted with a subscript to indicate which ion we're talking about. The Nernst potential is reached when the system is at equilibrium, so you'll sometimes hear it referred to as an equilibrium potential. The reversal potential is the point at which the net flux of ions switches directions. We usually refer to reversal potentials when talking about ion channels. Just to round out the terminology of potentials, it's worth recapping two other terms that you might have heard before: membrane potential and resting potential. The membrane potential is a much more general term that simply refers to the electric potential measured across the membrane. The resting potential is just the measured membrane potential for a cell that's at rest, that is, not actively sending a signal. For a neuron, the resting potential is close to the Nernst potential of potassium, but it's important to know that the two are not the same. The resting potential is composed of the flow of multiple ions, as well as the work of active ion pumps that we'll talk about later. Meanwhile, the Nernst potential is just the passive equilibrium for a single ion. We'll talk more about this difference in greater detail later, but it's important for now to know your way around the terminology. Let's now go back to the parts of the Nernst equation. The Nernst potential is equal to the universal gas constant times the absolute temperature, measured in degrees Kelvin, divided by the valence of the ion, times Faraday's constant, all multiplied by the natural logarithm of the concentration of the ion outside the cell, over the concentration of the ion inside the cell. Now, the universal gas constant and Faraday's constant are numbers you may have encountered if you've taken chemistry or physics. For our purposes today, you don't need to know anything about them, beyond the fact that you need to plug them in the right place. Let's go through some questions to learn how this equation works. Take temperature. Most warm-blooded mammals, such as ourselves, have a body temperature of around 37 degrees Celsius. What would be the change in the Nernst potential for an ion if we moved a neuron from 37 degrees C into a refrigerator at 4 degrees C?